Thanks, and good evening. So here you see an image of 3 cytotoxic killer T cells. They are surrounding and attacking a tumor cell in the middle. And I will return to this situation, and I will tell you a little bit of why it is important to understand and be able to control a situation like this. But first, control. So in order to be able to have any idea about control, we have to be able to make maybe some predictions or even about the future, which is hard, or even influence the future. And for that, we need models. And of course, what is a model, right? If you're a biologist, you might think about something like this. That is an animal model, an in vivo system where you can study some aspects of some biology that you might be interested in. But if you're a mathematician, you will think maybe of some system of equations or some other type of mathematical model where there are parameters that you need to estimate or fit according to measurements that you make on the system you're interested in. Of course, more advanced, or let's say more, during the last decade, there has been a lot of efforts into ex vivo systems, like this organoid system here. So organoids are 3D structures, biological structures that you can build that can mimic, for instance, a human tissue. So having many of the same cells that you see in the human tissue present and functional for days or weeks or months. And we can try out, for instance, different drugs and see how this tissue respond. And that is, of course, important because a mouse is a mouse. And if we are interested in biomedicine, we want to understand how humans would react to an intervention or a drug. And drug development traditionally goes that first we try things in vitro. So we have cell lines where we try different medications or drugs. We think about it, work on it, and then we move that into an in vivo phase where we maybe try it out on mouse models and see how that works, and then maybe move it even further into other animal models before we ultimately maybe test a few of these things that cross all these difficult phases that we ultimately can then try on humans, right? And of course, more often than not, what works in a mouse does not work in a human. Sometimes it does, but more often than not, it doesn't. And on the other hand, you might imagine that things that we stop because it does not work in a mouse might have worked differently in a human. And of course, therefore, things like organoids are important. And it's important to try to integrate all these different types of models and make them talk to each other, so that what we learn in a computational model or mathematical model can be fed over to what we then learn in an ex vivo system like an organoid. So ultimately, arguably the best model for human health and disease is human health and disease. This is the ground truth. What is a human? I mean, the latest estimates are that each and every one of us, we are composed of 30 trillion human cells, 30,000 billion, 3 times 10 to the power 13, if you prefer, human cells. On top of that, we have as many bacteria in our body, around 10 times as many viruses in our body as we sit here, stand here, and this is under normal, healthy conditions. Right? And it's very dynamic. During my 15-minute talk here, I will have almost 4 billion of my human cells will die on stage. Fortunately, almost as many will be born. And this is only telling you a little bit about there's dynamics here. All these living entities in our bodies, they respond to the rise and the fall of the sun, the circadian rhythm. There is periodicity here, until ultimately, of course, in the end, everything collapses. And there are many different scales, of course. If you look at a human cell, it's tiny. It's around 10 to 50 micrometers. A bacterial cell is much smaller. It's 10 times smaller in length. That's 1,000 times smaller in volume. A viral particle, on the other hand, is 10 times smaller still. 1,000 times smaller still in volume. Okay? So, and then all of these living entities in our body, they signal to each other. They talk to each other all the time, constantly. They are individual living entities, right? And they send off molecules, small molecules or larger molecules like RNA and DNA and protein. Our human cells, they have proteins sticking out on their surface, and they shake hands with each other, and that triggers programs, internal programs in the cell, and sets it off in different directions. And this happens all the time. So, in order to, let's say, model a human, we need to respect all the different layers here, how molecules, billions of molecules that makes up cells, and then all of these cells that makes up tissues, and tissues that organize into organs, that organize into organ complexes, that organize into an individual. And we organize into populations, and we interact with other organisms. And often, if we want to understand what is happening to an organism, a human, for instance, under a certain condition, we need to integrate information from many of these different layers in order to have a chance to have any solid predictions. Now, all our human cells with nucleus, they contain the same DNA. And the DNA, as you know, is the recipe for that is being transcribed to RNA, that's being translated to proteins, and proteins, they are doing all the interesting stuff. All the action that goes on in life are done by proteins, right? And still, they have the same recipe inside. So why do they look different under a microscope, for instance? We have bone cells and fat cells and neurons, nerval cells, etc. And they all look different, and they have different activities. And that is because they talk to each other all the time, and they talk to each other during development. They respond to their environment during the development, from a naive little cell to a mature cell doing its stuff. Right? So parts of the DNA is shut down and some other parts are activated, and that makes all different families of proteins that are responsible for having those different cells, like immune cells. So, of course, we have all these viruses and bacteria, etc., inside us under normal, healthy conditions. But sometimes we get infected by foreign pathogens. But we have a defense. So billions of white blood cells, immune cells, are circulating in our blood and in our lymph system, and they are continuously guarding us. They are looking for these intruders, viruses or bacteria that could come, and the first responders is the innate immune system. So neutrophils and macrophages and dendritic cells that immediately come minutes or just an hour after an infection. They are there to engulf and maybe eradicate the infection. But they are also sending off warning signals to the adaptive immune system, the T cells and B cells that are highly specialized and that can come into action if needed. And they clonally expand, and they are trained to hit a specific pathogen. And after they have successfully eradicated that pathogen, there is memory with T cells and B cells. So next time we experience the same pathogen, we are in a better situation. So let us come back to this. This is not an external invader that messes us up. This is our own cells that go rogue. So in cancer, a cell is normally mutated, and this mutation leads to proliferation that is unnatural. So it grows more than it should and continues to divide and grow and form a tumor. But that is due to a defect, a mutation, and that mutation shows also on the surface of the cell in terms of proteins that look slightly different. And actually, this is what the T cells should see, and they should then eliminate and kill those foreign cells. There is a model for this, the cancer immunity cycle. And this you should read as number 1 down there, release of cancer cell antigens. If a tumor starts growing, there will be cancer cells dying. Then the innate immune system will see them. They will pick them up, dendritic cells, cancer antigen presenting cells, move them to the lymph nodes where the T cells are sitting ready to recognize their particular antigen. If they do so, they clonally expand, they build an army of exact identical twins that then can move in the bloodstream to the tumor, infiltrate the tumor, and kill the tumor cells. And this will then eliminate the cancer. And this works under normal condition, but sometimes it doesn't. And then the cancer forms and the cancer continues to grow. And over the last decade, we have a new tool to fight cancer. We have immunotherapy. So the Nobel Prize in 2018 was awarded for something called the checkpoint inhibitors. And checkpoint inhibitors are, again, molecules sticking, let's say checkpoints are molecules sticking out on tumor cells and on T cells that shake hands. And by shaking hand, the attack of this T cell is shut down. And the idea by Honjo and Allison was that you can block this signal. And they developed monoclonal antibodies that could bind and actually stop this interaction, which then had the effect that T cells could do their work and kill the tumor cell. And this has transformed cancer care. So for some patients that before cancer immunotherapy, there were no options left, they were terminally ill, they are now cured. Unfortunately, it does not work for all patients. And the big holy grail now in cancer immunotherapy is to understand why it works for some patients and why it doesn't work for other patients. So response and resistance to therapy. And what we have in our toolbox are measurements. We are now doing a lot more measurements than ever before. We have our organoid models, we have our computational models, but we also have the ground truth, we have patients. And we can now, in a tumor tissue sample, we can actually do single cell analysis. So we can look at hundreds of thousands of single cell. We can see where the cell came from, what it's doing in terms of RNA expression, what it's doing in terms of protein expression. So we can read and listen in to this crosstalk by the cells in the tumor. And based on this, we hope to be able to, by mathematical modeling, map out the cancer immunity state space to understand where I, as a patient, where I am in this space, if I'm approaching a more healthy state or not. This is the hope we have. And of course, to finish off here, I mean, clinicians, they have always looked at patients, measured some things, and tried to cure. And this will not change. What is changing is that we have much more data, much more precision in doing our predictions of a certain intervention will work. And we have then the ability to individualize the treatment really based on all this data. To do that, we really need this human-machine partnership. There is so much data that you as a human cannot interpret, not alone. So we need a tool. We need digital assistance, and you cannot, it's impossible to not think about when Kasparov lost in '97 against Deep Blue, and where we said that this was the first time that AI could do intuitive stuff better than Grandmaster. And now, I mean, you have, of course, all of you have already written this talk by using ChatGPT, right? Just feeding in some of the keywords. And of course, things like this will help. But in order to achieve the goal of really understanding and modeling cancer, for instance, we need a joint effort by humans, and it has to be multidisciplinary. This will not be done alone by computational scientists or mathematicians or biologists and immunologists. We need to work together. And I think in the interface here, we need to have this human-machine partnership, really take the benefit from the best of two worlds. And it would be pretty nice, so this is a call to action, right? It would be pretty nice if we down the road from here, together, jointly, all of us could say that, well, you can pretty much say that we cured cancer. Thank you.